1. The life cycle of a keratinocyte. From birth to shedding, keratinization is the 28 to 40-day journey of a cell from the basal layer to the skin surface, transforming from a living, dividing cell into a dead, protective scale. The 3 phases. The cycle consists of mitosis, cell division, differentiation, morphological and chemical changes, and exfoliation, desquamation at the surface. Structural integrity. The primary goal is the creation of the stratum corneum, a bricks-and-mortar barrier where keratinocytes become tough and nucleated corneocytes. 2. Stratum basale, the proliferative engine. The basal layer serves as the skin's germinal center, where stem cells divide asymmetrically to maintain the population while sending daughter cells upward to begin differentiation. These cells express keratin 5 and keratin 14, providing a flexible internal cytoskeleton, and are anchored to the basement membrane via hemidesmosomes. Once a cell loses its attachment to the basement membrane, it stops dividing and begins the transition into the stratum spinosum. 3. Stratum spinosum, the synthesis hub. The prickle layer, named for its spiny appearance under microscopy, caused by desmosomes, cell-to-cell bridges that resist mechanical stress. Keratin shift cells stop producing K5, K14 and switch to K1 and K10, which are larger, more robust keratins specialized for terminal differentiation. Involucrin production. Synthesis of the cornified envelope starts here with the protein involucrin, acting as a scaffold for the cell's future armor. 4. Stratum granulosum, the transformation. Keratohyalin granule cells fill with dark granules containing profilagrin, which will later be processed into filagrin to aggregate keratin filaments. Odland bodies. Also known as lamellar granules, these tiny sacs secrete lipids into the extracellular space to create the skin's moisture barrier. Organelle degradation. In a process of programmed cell death, cornification, the nucleus and mitochondria are digested to make room for dense keratin. 5. Stratum corneum, the final barrier. The result is a stack of flat, dead corneocytes filled with cross-linked keratin and surrounded by a hydrophobic lipid-rich cement that prevents water loss. The surface maintains an acidic pH, 4.5 to 5.5, critical for the enzymatic degradation of desmosomes that eventually triggers invisible shedding. This layer acts as the primary interface with the environment, providing mechanical strength and biological defense against pathogens. 6. Regional variations in disease states. Palmoplantar skin includes a stratum lucidum and specialized K9 keratin for friction, while facial skin has a much faster turnover rate and smaller cells. In disease states like psoriasis, the cycle accelerates from 28 days to 5 days, causing nuclei to be retained at the surface, parakeratosis, and visible scaling. Model organisms like mice have helped identify that K1 and K10 mutations lead to fragile, blistering skin and hyperkeratosis. When things go wrong, hyperkeratosis, epidermolytic ichthyosis caused by KRT1 or KRT10 mutations, leading to fragile cells that blister easily and eventually form thick, warty scales. Palmoplantar keratodermas, specific genetic defects in K9 or desmosomal proteins lead to massive thickening of just the hands and feet. 8. The genetic blueprints, KRT1 and KR10. These genes provide the structural stability of the suprabasal layers. Mutations cause mechanical instability and blistering. Filagrin FLG, loss-of-function mutations in FLG are the primary cause of ichthyosis vulgaris and a major risk factor for atopic dermatitis, eczema. TGM1, transglutaminase. This gene encodes the glue enzyme that cross-links proteins. Its absence causes lamellar ichthyosis, where skin cannot shed. 9 insights from model organisms. Transgenic mice knockout mouse models for K5-K14 were the first to prove that keratin defects cause the skin blistering seen in human EBS. Zebrafish studies. Transparent zebrafish embryos allow scientists to watch keratin filament assembly in real time during early development. Evolutionary conservation. The hard keratins of hair and nails evolved from soft epithelial keratins, as seen in the transition from amphibians to reptiles. 10. Summary and clinical impact. Balance is key. Healthy skin requires an exact balance between basal proliferation and surface desquamation. Therapeutic targets. Retinoids work by modulating these genes to normalize the keratinization rate in conditions like acne and psoriasis. Future directions. Gene editing CRISPR is being researched to correct KRT10 mutations in patches of skin to treat congenital ichthyosis.